Many early medtech ventures must navigate uncertainty and small sample sizes. How do you balance statistical rigor with the need to be agile and responsive in early clinical stages?
Well, I think we have to recognize that the primary endpoint of any early stage study is whether you want to invest in a future study. So I think that you should not get too hung up on the p-values, and you should look at the data. You should look at the data in totality, and once you think that you've seen something, then I would apply the statistics afterwards and say, well, what I think I've seen, is it statistically significant? And ultimately, I mean, who is the p-value for? It's part of the tools that we use to convince people that there might be something interesting to pursue in these early phase trials. But by the very nature, I think early phase trials are underpowered, and for that reason, I would hate to see a small clinical trial, a novel idea with a large impact, abandoned just because it didn't achieve a p-value. We've heard during this conference that there's ways of trying to increase the power of your study, and I think if possible do a crossover design so you can compare the treatment. The patient as their own control is ideal, but for many interventions, of course, that's not possible. The other possibility is to make multiple measurements so that you get several baseline measurements to get a very secure baseline, and then do serial measurements after the intervention. And very often at one time point it won't be significant, but if you actually measure 3, 4, 5 time points, you'll find that cumulatively you've seen a difference. But ultimately, I think it is about trends, about looking at the magnitude of benefit, and not to get too hung up on the p-value.
And pharmaceutical drugs usually act via well-characterized biochemical pathways, whereas device effects can be more mechanical, systemic, or even unclear. In your opinion, how should trial design adapt to reflect this difference in mechanism?
Well, I sort of disagree with you. I think that we're equally in the dark regarding pharmacological mechanisms as well as device mechanisms. Ultimately, I think you have a hypothesis, you have a pathophysiological pathway, and you're trying to target that. You should be looking at surrogate endpoints to show that you've got target engagement. And then once you've got target engagement, then you're looking for some evidence of clinical benefit. And again, that might just come from surrogates, and my specialty is heart failure, so we're particularly interested in things like natriuretic peptides, but also kidney function, also things like ejection fraction, etc. Fundamentally, I don't think there is a big difference between the pharmaceutical and device industry when it comes to trial design, when it comes to the difficulties of interpreting early phase data. I guess the big issue with devices is the numbers of patients that you treat in these early phase trials is often in the 10s or 20s or 30s. Phase 2 randomized trial of a pharmacological agent, you're very often talking about 200 or 300 patients, so there is a difference in size in that respect.
And reflecting on your time shaping clinical trial infrastructure in cardiology, what one piece of guidance would you most strongly offer to founders planning to run their first device trial?
So, the one piece of advice, well, look, I guess the one piece of advice is to make sure that you have good people around you giving you advice, that you make sure that they're not just yes men or women, that you have people who are critical and will make you think. You have to have the courage of your convictions, of course, and to make sure that you collect all of the information, and very often that information is not expensive to collect. You just have to think about collecting it. And remember that, especially for conditions like heart failure, it's about how patients feel and not just about how much longer they live. So always ask the patient is the customer, and the customer is always right.
At this summit, we have a hybrid model where we have both innovators and investors attending, but also academics, and have a good volume of academic content. As an academic yourself, did you find value in this environment, and how does it compare to some of the other conferences you attend?
Well, this is a pretty unique experience to have all these different elements together, and a unique opportunity to see all of these innovations lined up, and also to get some feedback from the investors, what they see as a benefit. So I think that you've created something which is quite unique, quite different from what we see at academic conferences, where often it's late stage research or epidemiological research, often research done by junior investigators. Here you have really senior people. This is the passion, this is their living that they're showing you. So a very unique experience to see the procession of innovations out there that really make you think, and how to integrate them into current clinical practice, how they might play together, some of these innovations, that they might be well matched and work together. So yeah, altogether I sort of thought I was coming here as an educator, but I also got educated, so it's great.